Hi, I'm James Verdier, and welcome to the American Institute of Biological Sciences Bioscience Talks, a forum for integrating the life sciences, where we discuss the latest bioscience publications. As a reminder, if you'd like to read more, point your browser to bioscience.oxfordjournals.org. And in our second episode, I'm joined by Dr. Robert Devlin, who's with Fisheries and Oceans Canada. His work here is focused on the environmental threats that could be posed by growth hormone transgenic fish. In particular, his goal is to try to figure out what might happen if these fish were accidentally released into the natural world. And before we get into the interview, it's worth noting that the strains he works with are similar to those developed and patented by AquaBounty Technologies, a company that's currently seeking approval to commercialize its operations and produce aquaculture salmon for human consumption. So without further ado, here's Dr. Devlin. All right, so I'm joined now by Dr. Devlin. Thank you very much for joining us.
My pleasure.
So just to get us started, I was hoping you could give us a little bit of history or background about these growth-enhanced fish. When were they first invented, as it were?
Certainly. Growth-enhanced transgenic fish actually were first developed in China and in the United Kingdom in the mid-1980s. So it's really been more than 30 years now since transgenic fish or genetically engineered fish have been in research environments. After several years in the late 1980s, genetically engineered salmon started to be produced. And these are the ones that are primarily being focused on for production situations now, and the main topic of our risk assessment research as well.
And are these fish currently in evaluation for human consumption?
Yeah, so our fish are not at all for commercial purposes. We've developed our growth-enhanced transgenic salmon for research purposes only, for risk assessment analysis and to understand the data requirements that are needed for a robust risk assessment. But there are other groups globally that are producing genetically engineered fish, primarily growth-enhanced ones, for commercial purposes for use in an aquaculture environment.
Okay, and so the fish that you're mainly looking at, those are similar to the ones that we'll eventually see in wider commercial production, at least prospectively.
The fish that we're working with primarily are coho salmon, which is a Pacific salmon, and also rainbow trout, which is in the same genus as the coho salmon. Both of those are what you'd say from a distant view, they're very similar to the Atlantic salmon that are being produced for potential commercial use.
So what is it that makes the transgenic fish, both those that you've worked with and those that are being developed for potential commercial use, different from the wild type fish?
So the main approach that's been taken to modify fish transgenesis for aquaculture purposes has been the insertion of modified growth hormone genes. So growth hormone is a very high level regulating hormone that modifies the metabolism and behavior of the animal in very profound ways. And it's a very highly regulated in wild type fish. It's only produced in the pituitary gland, and it's expressed in controlled ways in response to environmental conditions, developmental profiles, and so on. And what has been done through the genetic engineering process is to take the gene for growth hormone and remove the regulatory portions of the gene that allow it to be regulated in a precise way, and to allow the gene, the modified gene, to be expressed in what one can almost call a dumb way, and that it just expresses growth hormone at all times and in many tissues in an unregulated way. This results in high levels of fish growth hormone in the animal and results in rapid growth. It's important to note that all the transgenic fish that are being produced for commercial purposes are only using fish growth hormone genes, and that particular hormone from fish is not active in higher vertebrates such as humans or other mammals.
So if I'm understanding that correctly, just taking an initial view, we wouldn't expect the enhanced growth hormone to have any effect in animals that might wind up eating the fish, including humans.
Potentially other fish might have some effects, but mammals and many other vertebrates probably not. There are secondary influences of the growth hormone on compounds in the animal, other hormones like insulin, like growth factor 1, and other chemicals that may have some effects. But our primary research has not been in the area of food safety issues. It's been focused on environmental risk assessment and what the potential effects of these animals might be if they were ever to enter the natural environment.
Okay, and what other differences do we see with the growth hormone fish compared with the wild type fish? Obviously they grow faster, but do they wind up larger, and are there any major behavioral differences?
Indeed, there really are. I mean, one thing we need to make clear is that the process, at least for salmonids, in general, is not making very large fish. It's making fast-growing fish that reach particular developmental stages in a shorter period of time. We do see some environmental conditions in the lab where the fish will actually get larger than a normal adult salmon, but in general, it's a growth rate enhancement rather than a overall body size enhancement. But indeed, many other characteristics have been affected. As I mentioned, growth hormone is very high level. It affects metabolism, which influences the division of cells and the requirement for energy. This in turn feeds forward to the brain to modify feeding behavior. The animals have what I would call a very voracious feeding behavior compared to wild type. Wild type fish are always balancing off the trade-off between the need to acquire food resources and to balance the risk when they do that of becoming food themselves.
So compared with the wild type, these are just very hungry fish.
They're certainly almost always hungry, and they will eat voraciously. And because of that, they really can have a very strong competitive advantage in environments where you produce or grow transgenic and non-transgenic fish together. The transgenics will easily outcompete the wild type fish. So that's one of the main concerns that people have had, is if these animals were to escape into nature, would they, their appetites be such that they would cause ecological effects through competition. But things are really not that simple for a risk assessment, because concomitant with the feeding behavior causing a competitive advantage, this feeding behavior also drives the fish to expose itself to predation risk to a greater extent. They're out foraging more. They don't hold together in a schooling or shoaling fashion, clustered together to minimize their individual predation risk. They're out and dispersed. And this leads to them having a higher level of predation susceptibility.
So, if they were to be released into a low predator environment, you might expect them to outcompete the native fish.
Indeed, if there is sufficient food, they certainly could have that potential. Whereas in a high predation situation, they likely would not survive. We've actually done experiments like that in naturalized stream environments where the transgenic fish are found to hatch out of the gravel and emerge into the freshwater stream sooner than will their siblings that are non-transgenic. And when that happens, the transgenic fish, in the absence of predators, have a distinct advantage.
And that would seem to be one of the difficulties with the accidental release of fish like this, is that it's probably, I'm assuming, very difficult to model all of the potential environmental circumstances that they might encounter.
That's very true. We have done some modeling. There's been some really excellent modeling over the decades by other groups that have focused on fitness-based influences, and these are primarily looking at the survival potential through different developmental stages and the reproductive potential, the two main components of fitness. And one can do some reasonable modeling in that respect, but the genotype by environment interactions, the plasticity of the animal and how it may change across environments has not been incorporated into those models as yet. But that's an objective for the future.
So essentially, even if you do have a reasonably good model, it's still not suitable to put your utter faith in it. You can't completely trust the thing.
I think that's the case for our transgenic salmon. We have quite a lot of data, but we certainly find incongruities in the information that we generate from experiments that only differ slightly in the environmental conditions. And so trying to make generalizations from those at this point is very difficult. The main thing that we found is just that there's a great deal of uncertainty in trying to take laboratory data and develop it into a robust set of information that a regulator could feel comfortable about making a firm and certain decision about the outcome. So I think uncertainty plays a role in all risk assessments, and in this case, unfortunately, the data that we've generated is largely variable, and therefore provides uncertainty for anyone using that information. And that's perhaps the main conclusion of the findings, is that we're not able to actually make a defined prediction about what would happen if the fish entered nature.
And one thing I wanted to jump back to something you'd said earlier. What's stopping us from doing, sort of a really simple, empirical test of an introduction? What makes this different from, some of the other genetically modified crop species that, we already have: corn, alfalfa, et cetera, where we've tested those. We know their invasion potential. Why can't we do the same thing with fish?
That's a very good question. I think the main reason, or there's twofold. One is the difference in the strains that are used as progenitors for transgenic fish are generally very wild type in nature. Transgenic fish made from those, if they escape into nature, would easily have the ability to breed and introduce the transgene into the environment. We've done those kinds of experiments in the lab, where we set up natural breeding environments and bring in fish from nature and transgenic fish from the lab, let them sort out who will mate with who, and certainly the transgenic fish do mate with wild type fish and do transmit the genes on to the next generation. So because of that, because of the generally wild nature of the progenitor strains, that's a big difference from many of the genetically engineered crops and agricultural animals, for that matter, that have been highly domesticated for perhaps many millennia, and we have a knowledge of what those organisms would do if they escape. If transgenic pigs escape from the farm, we're likely to be able to find most of them, and we also know what the history of those animals would be in terms of their potential for survival in nature. And that brings us to the second point, which is that the environments in which plant and agricultural animals are being raised in is very different from the aquatic environment. If transgenic or any fish escapes into aquatic environment, it's very, very difficult to ever try and recover them, certainly at high levels. So once a transgenic fish is out, there's very little you can do about it, and the consequences of its entry will ensue.
So, what kind of means are available to protect against that sort of accidental introduction, or having the fish flourish if it were to escape?
Yes, this has been well recognized for many decades now, that these kinds of risks are there. We now have lots of empirical data to show the uncertainty, and so there has been a focus for at least 15 years to develop two different approaches to trying to prevent this from happening. And the first, of course, is to develop high quality land-based but physically contained facilities where you can raise these animals with minimal risk of entry into the natural environment. So make your rearing environments escape proof. One first wants to focus on the physical containment side of things to make sure that the fish stay on land. But in the case that you want to go further, there's been also a focus on developing biological control methods. And the main one that's been explored for transgenic fish is to induce a condition called triploidy, where the animals have 3 sets of chromosomes rather than 2. And this results in the blockage of the proper development of the testes and ovary in these animals, rendering them functionally sterile. And so that land-based control plus sterility seems like a very good way to go in terms of having a backup system. Of course, for any containment system, it's very critical to know the efficacy of that. And for triploidy, we find that typically more than 98% of the animals are sterilized, often 99.8%. And that seems like a very high level, and it certainly is for the potential escapes of fish from a land-based facility. But if one considers the use of animals outside of those environments, for example, in net pens, in typical aquaculture environments, even a failure rate of 0.2% would still potentially allow quite a number of fertile transgenic animals to be released to nature. So we really need to improve the efficacy of containment methods if we want to consider growing these animals outside of contained land-based facilities.
So it sounds like there is a good program for containment, but just thinking speculatively, what if that containment were to fail and you did wind up with, a fairly large number of transgenic fish that entered the aquatic environment? What sort of effects would we expect, or do we just not know at this point?
Well, that's the key question. So the first question is whether they will survive in natural environments, and all of the simulations that we've done. I mean actual experiments in naturalized environments indicates that the transgenic fish can survive in naturalized conditions in ecosystems. Under different conditions, they may have enhanced survival or reduced survival, but they will survive, and they can survive to the extent where they'll grow up to adulthood and reproduce and produce transgenic progeny of their own, either by interbreeding with themselves or by breeding with wild type individuals in the same environment. And so the key question, though, is what's the relative fitness of these animals compared to wild type? So if they have an enhanced ability to survive and an improved ability to breed relative to wild type, the transgene will persist in nature and not be purged from the system. Whereas if there's a detriment caused by the transgene, of course, the transgene will be purged, and the consequences to the ecosystem will only endure for the time that that purging occurs from the population, and that will, of course, depend on the fitness impairment that the animals are receiving.
So it sounds like this puts regulators in kind of a tough spot because, in a sense, they just don't have enough information to make a firm decision, do they?
Well, they can make decisions,
Sure.
but they're taking into account, at least in Canada, the uncertainty that's unfolded from the empirical data that's been generated. And I think for us to try and come up with certainty for some of the strains, it will be very difficult. There could be some transgenic strains that are very obviously impaired in terms of their swimming ability or disease resistance, that they still might be suitable in sterile, land-based, or other environments for aquaculture, but the data will be so obvious that that they wouldn't make it in nature, that a firm regulatory decision could be made based on those. But the fish that we have developed here are very close in their characteristics to wild types, except for these changes in behavior and metabolism. So to have a certain prediction about what they would do in nature is difficult at this point.
And given that uncertainty, and I realize this question may be unfair, but I think it's one that our listeners will be interested in. What's the worst case scenario? Are we looking at extirpation of the native species, or are we looking at massive interbreeding, a large scale change in the aquatic environment, or do we just not know?
I think we don't know the answer, but I wouldn't anticipate extirpation of the species. One of the sets of data that we've generated is that the transgenic strain is very likely to evolve once it enters a natural environment and experiences different genetic backgrounds. As you know, wild populations contain variation, and they're constantly selecting that variation in response to shifting environmental conditions to to adapt to those conditions. And transgenic fish will be no different, and they will try to improve their fitness by selecting variation. And so I think it's unlikely that the transgenic fish would actually be able to dominate and breed with wild fish to the point where we'd see no non-transgenic fish. I think the non-transgenic animals would likely mount a counter response in terms of evolution, but until we actually know firmly what the fitness effects of the transgene are, unfortunately, it is speculative at this time.
And that leads me to my last question, which is, what's next for your research? Are you doing more ecological modeling and more threat modeling, or have you kind of moved on from that?
Yes, I think there's a little bit more ecological work to do to finish off the studies, some containment research that is underway, but we've shifted largely from pure transgenic research more to examining other types of genetically modified organisms, for example, those that have been created by selective breeding or domestication. It's interesting that for transgenic fish, we actually know precisely what the transgene is that we've inserted into these animals, whereas domesticated fish, which also can grow very fast, we don't really know the genetic basis of that, and we need to understand that to understand what their potential effects might be if these animals escape and breed with wild fish.
That sounds like a fascinating area of future research. Thank you for joining us, Dr. Devlin.
My pleasure. Thank you very much.
And that concludes this episode of Bioscience Talks. Just a reminder, the journal Bioscience is published by Oxford University Press on behalf of the American Institute of Biological Sciences. To read the article we talked about today, point your browser to bioscience.oxfordjournals.org. Thank you for listening. Talk to you next time.